in session 322, Hurthle cell thyroid cancers. These are not papillary or follicular thyroid cancers. Where is the research going in this disease and what has been done so far? And our presenter today is Dr. David McFadden, and I'm going to read your bio here. David G. McFadden, MD, is an endocrinologist at UT Southwestern Medical Center in Dallas, Texas. Dr. McFadden is an assistant professor of internal medicine and biochemistry. His clinical interest is in the treatment of tumors arising from endocrine glands, including thyroid cancer. He's a member of the American Thyroid Association and the International Thyroid Oncology Group, and has been awarded research grants from the National Institutes of Health, American Thyroid Association, and the Disease-Oriented Clinical Scholars Program at UT Southwestern, and the Cancer Prevention and Research Institute of Texas. His current research focuses on using genetic engineering and chemical screens to discover new treatments for cancer. I'd like to welcome you, Doctor, and welcome everyone.
Awesome. Thank you very much for the invitation and the introduction here. And to everybody out there, I'm David, and I'm going to talk a little bit today on an update on Hurthle cell tumors that come out of the thyroid gland and what a little bit of background, what we know, what we don't know, and kind of what we're working on as a field. Okay, I don't have any disclosures related to anything that we're going to talk about today with respect to conflicts of interest. Hurthle cell tumors are kind of an anomaly. There has been debate in the field over many years about where these tumors fit in the context of other forms of thyroid cancer. And if we specifically think about the two dominant forms of differentiated thyroid cancer, we think about follicular tumors, follicular carcinoma, and papillary thyroid cancer, or papillary thyroid carcinoma. And we know that these two kind of dominant groups of well-differentiated forms of thyroid cancer behave differently. They grow differently, they spread differently. Most follicular carcinomas, or follicular tumors, I should say, turn out to be benign. So they're not carcinomas, they're adenomas. These tumors don't spread to lymph nodes. And in the subfraction of cases that do spread, it goes to lungs or bone. It spreads through the bloodstream, not through the lymph nodes. And we know, we've heard a lot about BRAF mutations in thyroid cancer. We know that these follicular tumors, the true follicular tumors, don't have BRAF V600E mutations. And when we think about scanning and imaging, we also know that most follicular tumors are not very positive on PET scans. And this is a sugar tracer called fluorodeoxyglucose that measures how much sugar tumors take up. And there's some studies that suggest how aggressive or how fast growing a tumor is. They tend to take up more of this glucose sugar tracer and show up on PET scans. And most follicular carcinomas really don't. Papillary thyroid cancer, on the other hand, does spread through the lymph nodes before it spreads anywhere else, typically. It's uncommon for papillary thyroid cancer to spread beyond lymph nodes, but we see it in patients, especially at the busy centers, and that it does spread to lungs sometimes. Now, we know BRAF V600E mutations are probably the most common mutation in papillary thyroid carcinoma, but like follicular cancer, these are not very positive on most PET scans. Now, occasionally, as the tumor spreads or becomes a bit more aggressive, we will see it on PET scans. So the question, one of the questions we want to talk about today is sort of where do Hurthle cell tumors fit in? Okay, so Hurthle cell tumors share some features of both of these cancers. So most Hurthle cell tumors turn out to be benign adenomas, like follicular tumors. However, Hurthle cell tumors can spread to lymph nodes in the neck, or they can spread to little areas within the neck. And so this is more like papillary thyroid cancer. Hurthle cell tumors can also spread through the bloodstream when they're frank cancers and go to lung and bone if they do spread, but they can spread to other sites as well. They're a little less common in follicular carcinoma. We know that BRAF V600E mutations are also very uncommon in Hurthle cell tumors, and that's kind of more like follicular carcinoma. Now, Hurthle cell tumors tend not to take up radioactive iodine. Most of the time they don't. I like to say to my patients, never say never and never say 100% because we're not that good in medicine. So there are exceptions to every rule, and occasionally you'll find a Hurthle cell carcinoma that does take up radioactive iodine, but it's uncommon, less common than either follicular or papillary for sure. And unlike those tumors, Hurthle cell tumors almost always show up on these PET scans. You can even have benign Hurthle cell tumors, little thyroid nodules that are Hurthle cell nodules that will end up taking up iodine, glucose on these PET scans. So the bottom line is these tumors don't seem a lot like either follicular cancer or papillary, maybe a bit more like follicular, and that's why they were thought to be a subtype of follicular cancer up until recently. Now, I should mention that this name Hurthle cell is also a subject of debate. And so you'll see different types of names for Hurthle cell tumors, and they're all the same tumors. So they'll be called Hurthle cell tumors, and you'll also see them called oncocytic tumors. And in fact, the official kind of World Health Organization pathology group has decided to rename Hurthle cell carcinoma as oncocytic carcinoma of the thyroid. So if you see that term oncocytic, it really means the same thing. Okay, so do some of these different features, the unique features of Hurthle cell tumors, impact patient care? And I would say the answer is probably yes. Minimally invasive Hurthle cell cancers, so when they just have a couple of areas of invasion that allow the pathologist to call it a cancer or a carcinoma, these tend to behave a lot like minimally invasive follicular carcinoma in that surgery is often enough to treat these cancers, and they usually don't come back. 90+% of them don't come back, it seems, from most studies we have out there. Because Hurthle cell tumors tend to spread in the neck like papillary, your doctor might choose to use neck ultrasound more for following you than if you had a true follicular carcinoma. Now, because of that positive uptake on PET scans, I tend to use PET scanning more often for my follicular cancer patients, my Hurthle cell cancer patients than other forms of cancer. Is in the setting when I'm worried that the tumor might have spread, I'll turn to PET scans for Hurthle cells. Now, also treatments that target BRAF mutations or RET mutations that some of you probably have heard about don't usually work for Hurthle cell cancer because they don't have the mutations in the DNA of those tumors that would cause these proteins to be important for the growth of the tumor. We do know that some Hurthle cell tumors have RAS mutations, so as more and more progress is made to develop drugs that target RAS mutations, these might have impact for patients who have a RAS mutant Hurthle cell tumor. So one of the things the field has been challenged with in the last several years is trying to figure out what underlies the differences in the behavior of these tumors. Why are these tumors different? And then once we understand that, maybe we can develop better therapeutic approaches that really go after the origin of why these tumors are different. So what I'm going to talk about over the next several minutes is a little bit about why these are called Hurthle cell tumors. In other words, what they look like under a microscope. What do we know about what we call the cancer genome of Hurthle cell tumors? And this is really changes in the DNA within the tumor that don't occur in the normal thyroid of the same patient. So these are the genetic changes that we think may contribute to the growth of a Hurthle cell tumor. And what are these implications of what we can learn from these genetic studies with respect to research, where the research is going and needs to go, and how that might impact patient care someday? Okay, let's talk first about what they look like under the microscope. So these tumors are identified by pathologists by having this kind of lacy, you can see these little white areas within the cytoplasm, and the cytoplasm is a little bit purpley colored here. And then this is the cytoplasm, this big area of the cell. And this is where the DNA is in the nucleus of the cell. And I'll talk more about that in a minute. And the nucleus of a Hurthle cell tumor also looks different. It has this prominent dark spot in it that we call the nucleolus. So sort of the center of the nucleus, basically, is very prominent. These, as I mentioned, are also called oncocytic tumors. And so just keep that in mind, depending on what your doctors are using to kind of call your tumor. It means the same thing, basically. Now, this is under a light microscope, kind of a classic microscope that you might buy on Amazon. etc., or I played with as a kid, for example. If you use a real sophisticated microscope called an electron microscope, then you can kind of see why the cells look more purpley and different here. And these little kind of blobs that we see here are actually a huge increase in the number of a part of our cells called the mitochondria. And I'll talk more about what the mitochondria do in just a minute, but this is a Hurthle cell right here, and you can see the entire inside of the cell is taken over with these big swollen mitochondria. Now, this is a normal thyroid cell right next to it, and you can see these little darker areas are normal mitochondria, and there's usually some of them in a cell, but not completely taking over the cell, like you see in the Hurthle cell. So what all are we talking about with these parts of cells? So normal cells and cancer cells throughout our body are really divided into different little pieces or little parts that do different functions. So we have the nucleus, that center of the cell that I showed on the slide a bit ago. And this is where our DNA is. This is where our chromosomes are. This is all the instructions to kind of make proteins and drive the functions of cells. This little thing that I showed that gets swollen and is all over the Hurthle cells is called the mitochondria. And this is where most of our energy, chemical energy that's important for how a cell functions, this is where it's all generated. Like our muscle cells to do movement, that takes energy. And so we use chemical energy to drive the contraction and relaxation of muscles, and other cells have to do produce energy to do everything they want to do as well. Now, this kind of purpley, lacy area in the cells is called the cytoplasm. And this is kind of the soup of the cell where everything's kind of floating around. And we have a lot of proteins and everything that sit here and drive, again, the function of the cells as well. So this is our DNA. This is our energy producer, kind of the power plant. And this is where a lot of the function happens in the middle of the cell, cytoplasm. And as I showed you earlier, that these Hurthle cells really become totally composed of this mitochondria, really taking over the soup of the cell. So the question is, why are there so many strange looking mitochondria in Hurthle cell tumors? And this is something that's been observed for a long time. This is back in 1974. This is a study from Mass General Hospital in Boston. Kind of the leader of the thyroid group there, Farah Malouf, studied and kind of described all these huge increase in mitochondria. And during that study, in this kind of awesome paper that was published, they raised the possibility that maybe they're full of mitochondria because they're not working well. So the cell's trying to make more and more and more mitochondria to overcome some defect in the mitochondria. Okay, what about the alterations in DNA in Hurthle cell tumors? What are they? What's been done so far, and how do they compare to papillary in Hurthle cell? So the way these studies are done is basically we examine the nucleus, the chromosomes and DNA between a normal non-cancerous cell in our body. These are going to show us the DNA that we've inherited. And then we examine the DNA and chromosomes in the cancer. So presumably we think cancer forms when cells divide, a normal cell divides and grows, and one of the cells inherits a cancer-causing mutation that kind of contributes to the origin of that cancer, whereas the normal daughter cell doesn't inherit that mutation. And then how we identify that is kind of what I was saying, is we examine the DNA in a normal cell and we examine the DNA in the cancer cell, and we look for differences between the two. And we say, aha, this cancer has this change, which I show as a little red asterisk here, and this tumor or this normal cell does not. Okay. Cancer-causing mutations are important. It teaches us about the biology of cancer. And if we understand the biology and genetics, this can impact treatment. And that's kind of borne out over the last few decades, because understanding these cancer-causing mutations in thyroid cancers made a difference. We've learned that there are RET mutations in medullary carcinomas, and that these have really led to the development of a series of small molecules leading up to these really amazing direct RET inhibitors that are used in patients with medullary carcinoma. We know there's RET fusions occasionally in papillary thyroid cancers, BRAF mutations, RAS mutations, ALK changes. These are all genes that appear to be very important for the growth of thyroid cancer cells. And each of these has led to the development of a small molecule drug that blocks the action of these proteins in cells and acts as a good treatment. Okay, what do we know about Hurthle cell tumors? We know that these cells, as I've drawn here and shown earlier, are full of these abnormal appearing power plants, these mitochondria. And so what do we know about the changes in the DNA? What are these two little asterisks? Can we find some cancer mutations? Okay, when we think about chromosomes, if you go to like 23andMe, these websites that are doing, and these companies that are doing more and more genetic profiling of individuals to look for variation amongst us in the population and so forth, where we come from. We have 23 chromosomes. That's the origin of the name, but we have 2 copies of each chromosome. We inherit 1 copy from our mom and 1 copy from our dad. And having 2 copies of these chromosomes kind of is important. Now, as I said, we inherit 1 copy from mom and 1 copy from dad. And we can, if we knew the genetics of our mom and dad, we would know which chromosome comes from which, because there are little changes. And if we think about DNA, it's a sequence of these 4 letters over and over and over again. And that sequence is what drives kind of the production of proteins and the uniqueness of our individual DNA. And in the case of cancer, these little genetic changes going from a T here to a G here can be the origin of cancer, one of these mutations in one of these cancer genes. But we can also trace that this chromosome came from mom and this chromosome came from dad. And that's what 23andMe and stuff is very good at, because they know, aha, your dad has a G, you have a G. So this G came from your dad and this T came from your mom. And when we think about chromosomes, one of the important things we want to look at in cancers is the total number of chromosomes, because some cancers have alterations in the number of chromosomes. As those cancer cells divide, they don't just make mistakes like T to G, they get alterations where one cell might get 3 copies of a chromosome and one might get 1. And as cells divide, it's very important to split up the chromosomes evenly. So we can look at this in our DNA by saying, okay, this is a representation of the DNA sequencing data from one of these cancer genome studies. We have chromosomes 1, 2, 3, 4, 5 going all the way over here to 23, which I didn't label. And we know that there's these variants across the population in our normal DNA. It's what makes us individuals. And if we map those, if this DNA is from a cell or a tumor or a tissue that has 2 copies of every chromosome, then we see something like this, because half of the sequences have the mom chromosome, half of the sequences have the dad chromosome, then the fraction of T versus G maps to about 50%. So we see a nice line at around 50%. And this is indicative of a cell or a tissue that has 2 copies of the chromosomes. Now, the differences between mom and dad chromosomes and chromosome copy number can be followed in cancer genomes. And if we look at normal chromosomes, as I just showed, they look like this when we look at these genetic changes. But if we look at a Hurthle cell cancer chromosome, we see it looks very different. We see a lot of the changes in the normal tissues between mom and dad totally disappear, and other ones go to 100% of the chromosomes. And what this tells us is 1 copy of this chromosome disappeared in the growth of this cancer, such that we have only a copy from our mom or dad. Now, we don't know whether 1 comes from the mom, 1 comes from the dad. We just know that there's 1 copy here, because these genetic changes in the normal tissue either disappear or go to 100% of the chromosomes. And what that looks like when we kind of visualize it is it looks like this when we figured out the Hurthle cell cancer genome, that Hurthle cells lose 1 copy of most chromosomes, except they never lose a copy of chromosome 7, and they basically never lose a copy of chromosome 20. So there's something on chromosome 7 and chromosome 20 that these tumors don't like to lose. But the rest of the chromosomes, these tumors evolve as they grow and divide. These tumor cells lose most of their chromosomes, which is very unusual in most forms of cancer. And this is just what we might call a plot or a heat map showing how common this is across Hurthle cell cancers. And this is a study that I published a few years ago with a lot of collaborators and colleagues at Massachusetts General Hospital, where I used to work and where I really started and did a lot of this work before I moved to Texas. And so what we see, blue represents loss of a chromosome, red represents increased copies, increased numbers of chromosomes. So for chromosome 1, we see most of the Hurthle cell tumors lose a copy of chromosome 1, lose a copy of chromosome 2, 3, 4, etc. We see 5 is often kept around, but some tumors lose it. 6 is usually lost, etc. So if we look at this patient's tumor, we see it's lost a copy of 1, 2, 3, 4, 5, 6, has 2 copies of 7 like a normal cell, lost 1 copy of every other chromosome except chromosome 20. So this is a real signature of the Hurthle cell genome, and this is being used more and more in diagnostics. So the molecular studies like Afirma and ThyroSeq, they look for these changes in chromosome, these losses of chromosome copies, and that helps them figure out if your biopsy of a thyroid nodule is really a Hurthle cell type of growth in the gland. So this has kind of had a diagnostic impact on the field. And I should say that we weren't the first to show this. Investigators in the Netherlands showed this in a number of tumors a few years before us, but it was something that really came out as a big signature of these tumors. Okay, we also studied the kind of the little asterisks, the changes, the genetic mutations basically between normal tissues and Hurthle tumors, normal thyroid. And we saw that there's a few Hurthle cell tumors that have RAS mutations. We saw some of these other things like P53, TERT promoter, and a gene called DAX that we don't fully understand yet. But these are kind of common mutations that occur in other tumors and other cancers. And these TERT promoter mutations may be a component of a tumor being more aggressive, and P53 mutations are probably along the same lines. Now, most cancer studies kind of stop there when they think about what are the changes in the DNA of a tumor. But we actually have 2 genomes in our bodies, in our cells. We have the nucleus that I talked about and kind of have focused on so far. And this is kind of the information storage center. It's where the 23 chromosomes are. It's a big genome, 3.2 billion letters across all the chromosomes. And as I said, we have 2 copies, 1 copy from each parent. But it turns out these power plants, these mitochondria, have their own genome too. Now it's much, much smaller than our main genome, our nuclear genome. It's 16,000 and change letters. And it's not just 2 copies, it's hundreds to thousands of copies in each cell. And this genome kind of drives the production, the instructions here to make some proteins that are important in generating energy for cells. And this genome is very interesting because it's inherited exclusively from our moms. We don't inherit our mitochondrial genome from our dad at all. Okay, so in our study, we just talked about a little bit about the chromosomal genome, the nuclear genome, and now we want to talk about the mitochondrial genome. So we found that, there's, in our sequencing study, not surprisingly, there was a huge increase in the number of mitochondrial genomes in a Hurthle cell tumor compared to normal tissue. And that makes sense because there's a huge increase in the number of mitochondria. The kind of mitochondrial genome went along with that. Now, the other thing we found is we found mutations in the mitochondrial genome, alterations in the mitochondrial genome of these cells, just in the cancers. So not anything you didn't inherit. These patients don't inherit those mutations from their mom. These are kind of alterations in that mitochondrial genome that occur as the normal cell becomes a cancer cell. So they're only in the cancer cells. And we saw that the mutations that are predicted to affect some of the proteins made by the mitochondria occurred in basically all of the copies of mitochondrial DNA in these tumors. And so that suggested there's something beneficial to the tumor about having these mutations in these energy-producing proteins. Okay, what does this all mean? What am I talking about? Energy-producing proteins in mitochondria, et cetera. So as I mentioned earlier, cells need energy to function, to grow, to divide. And so a cancer cell needs a lot of energy to keep replicating, duplicating itself, and causing a tumor to get larger and larger. And cells get a lot of their energy from sugar. Okay, sugars we eat are broken down into simple sugars, and those are taken up by cells and broken down through a sort of a beautiful series of biochemical reactions into smaller pieces that are used to make energy. And we have a molecule in our cells called ATP that's kind of the main form of chemical energy that a cell has. And then these little pieces of sugars are broken down into building blocks that are used to make proteins, to make fats, to make other carbohydrates and membranes and components of the cell that execute the functions of the cell. And there are 2 major pathways that cells use to take sugar, and I'm going to call glucose kind of a simple form of sugar that cells really break everything down to. There's two pathways that cells use to take glucose and make energy. Okay, there's a kind of a pathway called glycolysis, the breaking down of sugar. This occurs in the soup of the cell, that cytoplasm. And this takes glucose and makes it into a smaller molecule called pyruvate. Now there's the function of the mitochondria, where this product of the first step, glycolysis, goes into the mitochondria. And there was a famous physiologist who described this other incredible series of reactions that take place in the power plant of the cell to generate energy from the sugar molecules. Now, what's interesting is if you break down glucose and just stop here, you get a total of 2 ATPs from the glucose. If you take that and run it through the mitochondria, you get 36 ATP. So you have a huge increase in the amount of energy you can get from sugar if you're using the mitochondrial pathway, the Krebs cycle. And so you can think about it, it's like money for the cell. You only get $2 if you run glycolysis. You get $36 if you take that same thing and go all the way through the mitochondria. So this mitochondria, this power plant, is really efficient at generating energy from sugar, and that can be beneficial to cells. And we typically think cancer cells want to be adaptive because they're growing, they're spreading, their nutritional needs may change. So keeping everything functioning would make a lot of sense. Now, it turns out that these mutations in Hurthle cells seem to do the opposite. They seem to totally impair, they're predicted to totally impair the function of that mitochondrial pathway. And so cells are really left with only being able to use glycolysis, these Hurthle cells, to make energy. And this seems counterintuitive, as I said. And so there's a lot of work in the field right now trying to figure this out. Why would a Hurthle cell want to kind of make energy generation less efficient? And we don't understand, and we certainly don't have all the answers to that. But it leads to the hypothesis that because these cells now are totally dependent on this pathway, that maybe if we can find drugs or small molecules that block this, that would have a huge impact on a Hurthle cell compared to a normal cell. And that's what we call a therapeutic window. We could find something that could be really detrimental to a Hurthle cell, but a normal cell has a way to adapt to that because their mitochondria are functional. And so this is one of the hypotheses in the field that's being tested right now. Okay, some summaries. And some caveats. So not every Hurthle cell tumor has these genetic changes in the mitochondria genome. We know about from the studies about 60% have these mito mutations that are predicted to impair the energy production. And we know that about 55–60% have the loss of chromosomes. So again, never say never, never say 100% in medicine. And it's not clear yet these mitochondrial mutations and the loss of chromosomes are actually crucial for the cell to continue to grow, for the tumor to continue to grow. We think they could be, but we don't know yet. And as I mentioned earlier, there are cases of Hurthle cell tumors that when it's metastatic or spreads to other parts of the body, that we saw some of those other mutations that are common in other aggressive cancers like TERT promoter or P53 mutation. And so there's an effort right now to understand the impact of these mitogenome mutations and someday possibly develop treatments that take advantage of this defect that Hurthle cell tumors appear to have in their mitochondria. And I can highlight a few kind of manuscripts that have just been published or been put online within the past year that I think are showing kind of the field is really interested in Hurthle cell tumors. And that there's a lot of effort involved in understanding these aspects. So this is a paper from Memorial Sloan Kettering, where they looked at metabolic changes, some of the consequences of the alterations in the mitochondrial genome and how that impacts the metabolism in the cell, but also the microenvironmental, got a little typo here, microenvironmental changes around the tumor as well. Another paper from Sloan Kettering showed that targeting this kind of central pathway in metabolism resulted in some preclinical models, so not necessarily taking it all the way to big clinical trials, but there's an effort underway for that. But there's some promise in looking at this pathway in Hurthle cell that's a big metabolic pathway. And then there are two papers, one from Massachusetts General and one from my group in Dallas, kind of beginning to look at this defect in energy production and what are the byproducts of that defect in energy production in Hurthle cell tumors, and how might we begin to take advantage of that from a therapeutic perspective. So just to tell everybody who's here, I assume you have some interest in Hurthle cell or oncocytic tumors. There's a lot of interest in the field. There's a lot of work underway. International Thyroid Oncology Group is really putting a lot of us who do this work in the same room. So we're talking to each other. We're coming up with ideas for research projects that could be collaborative, but we have a lot of work that still needs to be done to take these ideas from theory to testing them in the laboratory to actually things that might translate to our patients and our patient care. So I have a lot of people to quickly think. People of my lab have a phenomenal student who's doing a lot of this work, and another team, a second student who's doing this work, as well as others that are supportive. I had a phenomenal clinical mentor who taught me everything I know about thyroid cancer at Mass General, Dr. Gilbert Daniels. And I've had tried to mention a few, but I want to highlight several scientists and physicians who've made a lot of contributions to what I talked about today. Ian Ganley and Jim Fagan at Sloan Kettering are real leaders in this field. I mentioned the Dutch group, Willem Korver and Hans Moreau, who studied the chromosome changes. There's an Italian group I failed to mention who also found these mitochondrial DNA mutations. Again, before we did, we weren't the discoverers of that. And that's the Giuseppe Gaspari group in Italy. And then collaborators with our projects at MGH before I moved to Texas, Gadi Getz, Kirsten Kubler, Vamsi Mutha, and Raj Gopal, who's leading a lot of those efforts. And then there's been a lot of generous funding for Hurthle cell tumor research. And without that kind of funding, these projects would not be feasible. So I'm happy to stop here and address any kind of questions and so forth in the Q&A. So thanks for everybody's time and your interest in this.
Thank you. It was wonderful and so informative. I feel like I need to go back and take a few biology classes. There's a lot of questions that have popped into the Q&A. Do you prefer to read them yourself? And
yeah, I'm happy to do that. All right. I can kind of walk through this. I'll just make sure I get the right order here. Okay. Question in the Q&A says, I had a recurrence of Hurthle cell cancer in my neck in some areas involving the vagus nerve and recurrent laryngeal nerves. And the question is sort of, is it possible that this tumor recurrence came from kind of spillage of the tumor at the time of initial surgery? And the question is, maybe some high-risk Hurthle cell patients should be offered external beam radiation following surgery or after the PET scans I mentioned were positive. And this patient has no cancer anywhere else as far as one can tell by a PET scan. So I think this is not thought to typically happen from tumor spillage at surgery. Now, generally speaking, we know Hurthle cell tumors spread within the neck. And we don't think this is due to surgical kind of breaking up the tumor, cutting it, and the cells kind of dropping off at the time of surgery, because we see it so frequently in patients who never had surgery. Patients will show up with Hurthle cell in these kind of interesting tissue areas. There's debate about whether the spread of Hurthle cell in the neck is through the lymph nodes, like papillary cancer, or whether it's some kind of soft tissue seeding, which is not fully understood how that would occur, whether it's from cells in the bloodstream or not, or probably not. It could be that the tumor takes over the lymph node so that the lymph node's not obvious. So I would suspect that's kind of fitting. The Hurthle cell in those areas of the neck fits with sort of the natural history of a Hurthle cell tumor to show up there already. Most thyroid cancers are not thought to spread from surgery. Now, there are rare examples of anaplastic thyroid cancer spreading from a needle biopsy and so forth, but that's its own beast altogether. We don't think most thyroid cancers spread from kind of tumor dropping off at the time of surgery. Now, the second part of this question is external beam radiation. That's another subject of big debate. And it's an individual decision. A lot of thyroid cancer experts, physicians at the big centers don't like to use external beam radiation, particularly often in patients, unless they know there's tumor left behind that needs to be taken care of and that iodine's not going to take care of it, which is frequent with Hurthle cell. But it's a real individual case-by-case basis. I think it's very important to review the pathology. And if the pathology showed little microscopic foci of tumor around the thyroid and left behind, then I think raise with your physician that possibility of whether radiation might be useful. But it's a tough decision, I can say clinically, to make the decision when patients will benefit from external beam radiotherapy and whether and when they won't. So it's a hard call, I would say. It is offered on select cases for Hurthle cell. It's definitely something that we use in our patients who have disease, tumor left behind after surgery. Okay. I hope that helps a little bit, even though I apologize for waffling, but we do such individualization of everything in these cases. So the next question is, do Hurthle cell cancers affect mitochondria elsewhere? Is, for example, the heart and lung. And so no, they're not thought to impact. The genetic changes we see in the mitochondria are only in the Hurthle cells. They're not even in the normal thyroid from which these tumors arose for the most part. So it's thought that the rest of our body's mitochondria are acting just fine. It's just the mitochondria within that Hurthle cell that are defective. Okay, and there was I think part of the origin of that question is in the second question, that a patient had spread of Hurthle cell cancer to the lungs and heart failure and kind of a defect in the conduction system, kind of one of these EKG changes called a left bundle branch block. And that's hard to say. You know, I would suspect that may be due to some other aspect of the tumor spread and not necessarily a genetic change in the mitochondria in the heart that was driven by the Hurthle cell tumors. Okay, how do you know how to treat, the next question, how do you know how to treat Hurthle cell? Had a parent diagnosed several years ago and has metastasis of the lymph node and lung. So Hurthle cell, as I hope is clear, is a challenge to choose treatments, because as I mentioned, unlike some papillary cancers that have BRAF mutations or RET mutations, or medullary cancer with RET mutations, or follicular carcinoma with certain changes, we don't have therapies directed to the underlying alterations in Hurthle cell cancer yet. So these tumors, though, do respond on occasion to some of these multi-kinase inhibitors that are approved for other types of thyroid cancer, like lenvatinib, for example, can exhibit responses in patients who have metastases. One of the challenges with any thyroid cancer patient is choosing when to bite the bullet and dig in and take one of these systemic therapies. So usually the kind of oncologists want the tumor to be growing significantly over a certain window of time, because a lot of patients will have slow growing tumors, and there can be a benefit of doing nothing sometimes, just watching with kind of repeat the scans every several months or so to watch the growth, follow that thyroglobulin. Most Hurthle cell cancers make thyroglobulin, so it tends to be a pretty good marker for following how fast cancers are growing, Hurthle cell cancers are growing. So, and so watching a long time can be a good approach. If a patient has a single metastasis that chooses to grow and everything else is quiet, then we sometimes do focal treatments to that one spot that's growing. That can be a focal surgical treatment. If one spot shows up in the neck and everything else is quiet, you can go address that and kind of take the growing area out. Other areas around the body that can be affected by Hurthle cell, if one spot is growing and everything's quiet, Sometimes patients will see a radiation oncologist who will do kind of focused external beam therapy to that one spot that's growing and kind of shut that down. Sites can, some places, it would be easy to do a surgery. Now, I hate to use words like easy in any kind of cancer. So it's not easy to have surgery, but it's easy for the surgeon to do the surgery and take that area out. So one of the themes with any patient with really any form of thyroid cancer that has spread is individual individual judgment, kind of individualizing the therapy to that particular patient, because thyroid cancer, including Hurthle cell, is definitely not a one size fits all. So depending on whether the tumors that have spread are growing, that can really drive whether other therapies are needed. And depending on whether one area versus multiple areas are growing, can also drive some of these decisions. Okay, we have a very insightful, interesting question here. Could intermittent fasting help reduce Hurthle cell cancer growth by reducing insulin-like growth factor, or insulin-like growth hormone? Could metformin or semaglutide be helpful in slowing growth? And would these agents be helpful to improve the efficacy of external beam radiation? The short answer is we don't know. In general, my suspicion is that a Hurthle cell cancer, even in a patient who has excellent glycemic control, is going to be able to get enough sugar from just the natural circulation of sugar to fuel its growth. But it is possible that if we had sort of blood sugar lowering treatments, maybe those could be a part of a regimen if we find ways to target this mitochondrial defect. Now, metformin is interesting. Metformin has the potential to slow down these mitochondrial proteins involved in energy production. But metformin slows it down, but these tumors have already kind of evolved with that whole thing shut down. So I don't think metformin by itself is going to have a major impact on Hurthle cell cancer growth. Maybe if we figure this stuff out and a certain diet gets used in cancer. And I know there's investigations of sort of dietary modulation across many cancer types. And there's been some studies of breast cancer based on the genetic changes in the breast cancer, whether that may be more or less responsive to diet modification in cancer. So I think this is an unknown area. It's a very interesting area, but we don't have clear answers yet. In general, I tell my patients, live healthy, eat healthy. That's certainly going to help in general your cancer care as well. But I don't right now put them on any specific diets or anything. But I recommend a general healthy diet. And probably a lot of us would benefit from avoiding sugar-rich carbohydrates, basically. Okay, next question. Have you ever seen Hurthle cell in parathyroid? So that's an interesting question. Now, I'm not sure exactly the origin of the question. So there are oncocytic, kind of Hurthle-like cells. Again, that sort of name change from Hurthle to oncocytic that's recommended by the World Health Organization pathology group relates to the fact that oncocytic cells that look like Hurthle cells with all these mitochondria arise in other tissues as well. And one of the tissues that a Hurthle cell, not necessarily a cancerous or even a neoplastic, a tumor form of Hurthle cell. Hurthle cells occur in Hashimoto's thyroiditis, but they're not a growth. Those are just a reactive cell type that occurs in the thyroid. Parathyroid can have Hurthle cell or oncocytic tumors as well. And so there's a lot of parathyroid growths, almost always benign, that can be oncocytic, that can show those features. Now, that's one aspect. So there are oncocytic growths of the parathyroid gland, and many parathyroid growths kind of show some of this. It's possible that a Hurthle cell cancer itself from the thyroid could invade into the parathyroid. I don't know if that was part of the question either, but a locally aggressive Hurthle cell can, because the parathyroids are right next to the thyroid or intertwined with the thyroid sometimes, they can be invaded and kind of come out when a patient has surgery for Hurthle cell. Okay. Another question, given the direction of research into mitochondria, what are the best treatments available today? Right now, there are no treatments that we have that target these genetic changes in the mitochondria. So we're still relying on the TKIs, the tyrosine kinase inhibitors, the multi-kinase inhibitors. There's work from Sloan Kettering looking at this mTOR pathway and those mTOR inhibitors. Those are clinically used in other cancers. So I think those can be used, and there are certain oncologists. I'm not an oncologist, but some of the oncologists that are treating aggressive Hurthle cell will use some of those mTOR inhibitors. So TKIs and mTOR inhibitors are really the mainstays of the systemic or chemotherapies that are used today. Another question, ablation for Hurthle cell, alcohol, or I'm assuming radiofrequency ablation. And I don't think we know, I don't think we have a definitive study there. We use alcohol sometimes. I think as RFA, radiofrequency ablation, becomes more adopted in the field, that may be used as well. But I don't have a clear answer on one or the other being better right now. It sort of depends on where it is. Certain areas, they're using more radiofrequency type approaches like liver or bone, whereas sort of cervical lymph nodes in the neck, sometimes they're using alcohol. But I know a lot of kind of interventional radiologists, interventional endocrinologists, and other surgeons are starting to implement radiofrequency ablation approaches there too. Okay, do Hurthle cells have a high tumor mutational burden or PD-1 levels that make them amenable to immunotherapy? Great question. Most Hurthle cell tumors don't have an exceptionally high tumor mutational burden. Depending on the study you looked at, Hurthle cell overall mutational burden may be a little bit higher than follicular carcinoma, for example. But it's not sky high like some of these colorectal cancers, melanomas, lung cancers that have very high tumor mutational burdens that predict that the tumor's making abnormal proteins that could be recognized by the immune system, particularly in combination with some of these checkpoint inhibitors like PD-1 or PD-L1 based approaches. So, but this is a growing area of interest. And there are questions out there on whether the metabolic aspects of a cell can modulate that immune microenvironment. And that was sort of in one of those papers that I highlighted at the end from Ian Ganley and Ed Resnick at Memorial Sloan Kettering. They kind of looked at the immune composition around Hurthle cell tumors, and there were some interesting changes there as well. I don't think we yet know whether Hurthle cell is going to be particularly susceptible or resistant to immunotherapy approaches yet, but there's a lot of active research in that area right now. And another question, do HCC have a high mutational burden? Okay, great question. So two folks are asking the exact same question. So hopefully I kind of address that. There's a lot of interest in this. Some of the proteins that are involved in metabolism may be involved in the T cells, the immune cells that play a role in immunotherapy responses. So this is again a growing area of research, and I think most of the time, what is happening is, if PD-1 or immune therapies are being considered, the pathologist will often look for expression of PD-1 or PD-L1 in the tumor histology to actually say that the high expression predicts a response to immune therapy. So that's always an option to have a look with the pathologists on the expression levels of these things.
We have just under 10 minutes remaining. And we would encourage people to put their questions into the Q&A box. And it looks like another one just popped in there.
Okay, so another question is, an oncologist and ear, nose, and throat surgeon treated a 2.5 centimeter Hurthle cell carcinoma encapsulated with surgery and radioactive iodine. And we're talking 15 years ago, and no recurrence to date. And the imaging is continuing to be PET scans and maybe RAI scans. And the question is, should I look for a different follow-up method? I mean, first off, after 15 years of treatment for a relatively small Hurthle cell carcinoma, I don't know the pathology, whether it had just capsular invasion to be called a carcinoma or vascular invasion. If it was just capsular invasion and you're 15 years out, the probability of a recurrence is exceptionally low. And again, as I said in my talk, I use the phrase with my patients, never say never, never say 100%, but nonetheless, it's exceptionally low if it were capsular invasion only. And even if there was a foci or two of vascular invasion, the fact that there's been no recurrence over 15 years means the risk is low. I would be using a thyroglobulin blood test, and I assume that's very low as well. I wouldn't probably be doing routine PET and RAI scans, but I don't know the full details of the case. And so I think you should feel a sense of reassurance that over 15 years, there's been no recurrence to date. And I'd be following the thyroglobulin. If the thyroglobulin level is undetectable or even low but flat, I would feel reassurance, and I would space out the imaging and do that periodically and less frequently over time in my patients.
Yeah, she did add that it was CAP, so CAP, I don't know what that word
Okay,
is.
capsular invasion. If it's capsular invasion only, even without radioactive iodine, there's probably a 7%, 5 to 7% recurrence rate based on some studies from Sloan Kettering. So, and the thyroglobulin's undetectable. Yeah, I don't know that I would personally do PET and RAI imaging every year necessarily. I would space it out a little bit and let that thyroglobulin kind of reassure me and guide some imaging, maybe an ultrasound of the neck every now and then if the thyroglobulin changes, you could decide whether to do other imaging tests.
Linda's got some commentary and then a question. She says her recurrence was 25 years later when she reduced hormone and her TG did detect it. And then her question is there any link to radiation exposure?
Yep. So, certain thyroid cancers are linked to radiation exposure. We know from Hiroshima in the World War II. We know from the nuclear accidents as well, that strong exposure of radiation causes genetic changes in cells. We don't know that that's especially true for Hurthle cell, because most of the tumors that arise from radiation exposures, classically anyway, in thyroid cancer, have these kind of chromosomal breaks and kind of putting back together of these chromosomes. That doesn't exclude the possibility that other genetic changes can be caused by radiation, but Hurthle's not one that we think is really driven by that classic radiation exposure. And then just a personal anecdote here, that thyroid cancers do recur late, and this was a patient who had 25 years after initial treatment. When thyroid hormone was reduced, thyroglobulin came up, and that picked up a latent tumor that decided to grow some couple decades later. And this falls into the never say never, never say 100%. And thyroid cancer is one of those cancers that has late recurrences over time. And that's why I encourage all of my thyroid cancer patients to be with me for the rest of their lives. Some I see every several months, some I see once a year, some I see every couple of years, based on this risk overall. Okay, let me see another question for metastases to lung, about 10 millimeters. What would be next in the line of treatment, radiation or systemic treatment? That to me really depends on the growth of that area. If the area is not growing, then I personally would watch it and let it prove that it wants to grow before doing a specific treatment. If that one area were to grow, I would not necessarily typically go down the chemotherapy route, the systemic TKI therapy. I would consider options for a focal treatment. That could be a focal surgery, that could be a focal radiation, and think about those options, discuss those options with my physician, my team of physicians.
They did list their growth there.
Yeah, 7 to 10 in a year. I would, again, all these are kind of individual cases that you got to be careful with stating strong claims on or strong recommendations on. I would discuss with your physicians the options, all of those options, whether to wait several months and take another look at it with an imaging study, or doing something focal. And if there's truly just one spot, I would consider the focal treatments definitely before doing a systemic therapy there. If there's no other known disease, a PET scan is an option here to see, to kind of get a sense of whether there's any evidence of surprises anywhere else. Hopefully not. And if there's really nothing anywhere else in a single spot, I would be debating between a focal type of treatment or watching this and seeing it over time. It does appear there's a little bit of growth, 3 millimeters of growth in a year. So I would be discussing those options, basically.
We have about 3 minutes left. There's no new questions. I have a question for you.
Sure.
Does Hurthle cell occur just as Hurthle cell, or could you have like classic papillary plus Hurthle cell, or classic follicular plus Hurthle cell, or maybe even medullary plus Hurthle cell? Does that happen?
Yeah, so there are occasionally what are called oncocytic variants of papillary cancer, or oncocytic variants of others. Now, oncocytic follicular carcinoma is really Hurthle cell. That's what Hurthle cell is. If it's a follicular tumor and it's oncocytic, that's a Hurthle cell tumor. So there are oncocytic variants of papillary. That just means they have these mitochondrial changes or increased mitochondria that are seen on by the pathologist. But oncocytic papillary is not necessarily any more aggressive or any different than regular papillary. Our treatment kind of algorithms are generally the same for the oncocytic variants there. And so I think it's a great question. And there are these oncocytic variants of different types. Mostly papillary is the one that we see the most. There's also research that may suggest that certain forms of papillary also have these mitochondrial changes. Now, there needs to be more research into that too, because it turns out thyroid cancer is one of the cancers in the body that has the highest frequency of these mitochondrial genetic changes that I discussed today. And it seems there's at least one study, and there needs to be more work on this to make sure that this is sort of true across centers and other groups of patients, that tall cell variant papillary may have an increased frequency of these mitochondrial changes. So the research into Hurthle cell may translate and may be applicable to other cancers that have these mitochondrial changes. They also occur in some colon cancers and some kidney cancers as well. So Hurthle cell is kind of a clean system in a way, as an experimentalist, to say, okay, these tumors have this a lot. It's pure in these tumors. We can study it in these tumors, and maybe it'll have application to other cancers.
Two quick last questions here, and we'll ask our host to please let us extend a minute. Do Hurthle cell tumors ever stop producing thyroglobulin? And does Xgeva have any secondary effect on Hurthle cell growth?
Okay. The first question, does a Hurthle cell occasionally stop producing thyroglobulin? Sorry, I want to get the answer is yes. So most Hurthle cell tumors tend to produce thyroglobulin, but not always. So, another phrase I use for my patients is no test in medicine is perfect. And so that's why we rely on a complementary set of tests. No imaging modality is perfect. And so Hurthle cell tumors will sometimes stop making thyroglobulin. And so we have to be aware of that. And one of sort of relating back to your question as well, that I meant to say is there are also sort of poorly differentiated Hurthle cell tumors. So we think when they start off as a Hurthle cell cancer, acquire other genetic changes, that they can become poorly differentiated. And there are rare cases of anaplastic cancer arising in a Hurthle cell tumor, but that's not frequent at all. It's not something people should be kind of worried about with a Hurthle cell cancer. And does Xgeva have any secondary effect on Hurthle cell growth? So Xgeva's denosumab. I had to double check that. I know my drugs by their kind of generic names and not the trade names. So that's why I was looking down at my phone a second ago. I wanted to double check on what Xgeva is. Xgeva is used in patients who have metastatic thyroid cancer because some studies suggest that metastatic thyroid cancer in bones, because these anti-resorptive agents that reduce the breakdown of bone seem to have benefits on patients with bone metastases across all forms of cancer. So if the tumor's in bone, then Xgeva is used because there are some studies suggesting that can reduce the complications related to a bone metastasis and potentially reduce the frequency of bone metastases in some of these cancers as well.
Okay, we are over time. So at this point, we need to say thank you. We really appreciate the presentation here, Dr. McFadden. It was wonderful and super, super informative. And I want to thank our participants for really fabulous questions as always. And wish everybody a great rest of the conference. And hopefully we see you in person next year.
All right, sounds great. Take care and thank you so much.
Thank you.
Bye-bye.
Bye-bye.